Hey everyone, Professor Davis here, and we're going to be diving into the world of microbiology. In these set of lectures, we're going to be looking at different concepts and topics related to microbiology, but this first set's going to be an introduction. We're going to look at the microbial world, but also how it relates a little bit to you. So let's get started. So before we actually dive into more of the core stuff of what microbiology is, let's look at some terminology that is important. The first thing that's really important is looking at the idea of what we call microbes or microorganisms. These are what we consider germs most of the time by people. Now, they're not all things that may potentially make us sick, but they do include things like viruses, bacteria, protozoa, and yeast. These are the main components that make up microbiology. Now, viruses are not classified as actual microorganisms, even though they kind of get put in this group because they are not considered living things. Microorganism, the idea of an organism means that it's living. These guys are not living, but they do kind of get put into this category because they're microscopic and they're small. So a question I have for you is, are all germs bad? Are all of these microorganisms bad? And the answer is no, they're not. A lot of them, majority of them, do not want to harm us at all. And a lot of them are actually beneficial to us in different ways. But the ones we learn the most about and we think of when we think of microorganisms or microbes are the ones who actually do cause disease. Now, if they do cause disease, they are considered pathogenic. So pathogenic means they're disease causing, and that is the part that we normally focus in on as humans because that's what hurts us. We will spend several videos going over different types of microorganisms that affect different body systems in us, from our skin to our lungs to our cardiovascular system like our heart, and we will look into those because those are the ones that are obviously more medically important, but we are going to look at microorganisms as a whole first. Now, before we actually dive in and look at the different groups, we need to look at something called nomenclature. Nomenclature is the naming and classifying of living organisms. Now, this is all living organisms, but we're going to focus in on the microorganisms. Now, remember, viruses are not part of this because they're not considered living things. So there are 8 basic taxons now in this naming system that we call taxonomy. These guys go from domain being the biggest, okay, so the biggest group, the most inclusive group, down to species, which is the most inclusive group. It's the one that only one thing fits into. And it's a series. We do see that it goes in a particular order, and I would highly suggest that you learn this order. It goes domain, kingdom, phylum, class, order, family, genus, species. Now, how are you supposed to remember this? Well, one way and one trick to help us remember a lot of times things that we're learning in like an order is to put together a type of mnemonic. So I have an example here that says, domineering King Philip came over for good soup. It's a sentence that has the beginning letter of each of the categories, and it helps me keep them in order. You can create one of your own. You can also look some up online. There's lots of different ways you can potentially put these in order. Now, when I ask you these questions, I'm not going to give you some weird words you've never seen. It's going to be these, but you're going to need to make sure that you know them in order. Now, the species is really important because that species name is what we call a binomial name. Okay, binomial means it has 2 parts to it. And so we call this a binomial nomenclature. This was developed by the scientist Linnaeus, and he came up with this. Now, we use Latin for the language here, and that's important because Latin is a dead language. It's not one that's evolving and changing, and so we utilize it so it stays consistent in biology. If we were to pick English, English is constantly changing. Words don't always have the same meaning after a while, okay, from one generation to the next. And Latin being a dead language, it's going to be where we can utilize it without it changing. Now, the species name is going to also be known as the scientific name of the organism, and it's always the genus plus what we call a specific epithet. Now the genus is always capitalized like you see here, and then the specific epithet is not. Okay, and so this is important because it's a two-name system. So here's an example. This is one of the organisms we will be looking at. This is the species Staphylococcus, which is the genus, and the specific epithet is aureus. Now, when we look at this, Staphylococcus aureus is the actual scientific name of that particular bacteria. Now, if you're handwriting this, you're going to underline it, but if you're typing it, you want to italicize it. Now, this is going to be important in future assignments, so make sure that you use the proper way of documenting the scientific name. Now, when we look at this, a lot of times the genus gives you some information. This one tells you it's Staphylococcus, which means that it's clusters. Staphylo means clusters, coccus means spheres. So it almost kind of looks like under the microscope, like a bunch of grapes all bunched together. But the aureus means that it grows golden, and its colonies appear golden when it's on certain plates, okay, when it grows on certain augurs. Now, the abbreviation of this can be done. So once it's referred to the first time as Staphylococcus aureus, we can actually abbreviate this with the S capitalized and a period, and then aureus spelled out. So that is the abbreviation that we can use for the scientific name as well. Now, taxonomy is a constantly changing field of biology and field of science because of the fact that we are getting new information all the time. And as we develop new information, we start to realize that things are maybe not as closely related as we thought they were. So that's what actually, for a lot of us who are maybe a little older, we used to know that kingdom was the highest. Now we have a domain, and the domain was put together because they realized that Monera, which was the kingdom of bacteria, and it contained all bacteria, was actually not accurate. And they separated that into two kingdoms. These two kingdoms are bacteria and archaea. Okay, so we have bacteria and archaea. These are both prokaryotic cells, cells without a nucleus. And then we have the eukarya group, which are the cells that have a nucleus. Our focus of this class is mostly with the group bacteria. Now, we're going to have a few organisms in the eukarya that we're going to look at that are in the kingdoms protozoa, fungi, and even animalia. But bacteria is going to be the bulk of our studies. And remember, bacteria and archaea are prokaryotic cells. Eukarya are eukaryotic cells. There are also 4 basic kingdoms that are found in the group eukarya. So coming from eukarya, we do see that there are a set of 4 kingdoms. This is protista, fungi, plants, and animals. We are focusing in on protista, fungi, and animals. Okay, there are no microorganism plants that we're going to be studying. We're going to be looking at these particular groups. So all the ones shown here in kind of the pink are our focal points for this study of microbiology. All right, so let's review again some more terms. These are terms you should have learned back in a general biology course, and so we want to just review them so then that way when they come up, you know, you remember what they mean. So first, cells. Cells are membrane bound, and they do contain DNA. Now, the DNA may be housed in a nucleus, or it might be in a general area that we call a nucleoid. Where the DNA is located determines the cell type. So there's 2 cell types. There's prokaryotic versus eukaryotic. Prokaryotic is when the DNA is found in a nucleoid region, and eukaryotic cells are when it is found in a nucleus. All right, so in this picture here, A is the prokaryotic cell. These are going to be the domains bacteria and archaea. And B is the eukaryotic cell, which is going to be the kingdoms protista, fungi, and animalia. Some other terms are going to be unicellular versus multicellular. When we're looking at unicellular, uni is a prefix that means one, cellular means cell. So these are organisms that are composed of one cell, whereas multicellular organisms are organisms composed of many cells. So if you look here, we do see that we have unicellular organisms. These are going to be things like amoebas, bacteria, parameciums. You're going to find these mostly in the group bacteria and in protista. In the other side, when we look at the multicellular organisms, these are going to be mostly like your plants and animals, and we're focusing more with the animal side. Now, fungus is not in this flow chart, but fungi would be part of the multicellular organisms too, except for some exceptions when we look at yeast. but we'll talk about those exceptions when we get there. Another thing we need to make sure that we understand is, how do these organisms get their nutrients? How do they acquire their food? So there's technically two different ways. There's the autotrophs and there's the heterotrophs. The autotrophs are the ones who are the self-feeders. They're the ones that can create their own food from resources that are provided, whereas heterotrophs have to actually consume their food. So if we look at this little kind of chart or picture, you see the autotrophs are the self-feeders, and they can do this either with light. That's what the photoautotrophs are. Those are with your plants, your plankton, your green algae, your cyanobacteria. They can be the chemoautotrophs, which use chemicals. These are going to be found in the deep ocean where light does not penetrate. Now, these guys are really ecologically important because they are a big part of the food chain. They're the base of it, actually, because they can utilize the nutrients straight from the sun. They're able to take that energy from the sun and convert it into something that other organisms can use. Heterotrophs, on the other hand, are going to be where they have to get their food from some other source. So they either have to consume it and ingest it and eat it, or they have to absorb it. And this is why they're normally called consumers. Now, these guys do have some ecological importance too, because some of our consumers are decomposers who recycle nutrients so that the producers or the autotrophs can use those. Some are also for medical significance because they do cause sickness and illness in us, because they're eating our cells, they're consuming our cells, so they make us sick. And then we do see that majority, though, are going to be beneficial. They don't actually hurt us or make us sick. All right, so these are some terms. We are now going to complete a chart. In this chart, we're going to look at all the different groups that deal with the microorganisms, and we are going to utilize a lot of these terms that we have just looked at. So this is going to be kind of a basic overview. Now, first, we're going to start with viruses, even though viruses are non-living. Okay, so viruses do not have a cell type because they are not considered living things. Now, because of this, they are known as being acellular. Anytime you put a in front of a word, it means without. So here it means without a cell. They don't actually have a cell. So how do they get nutrition, or how are they even able to replicate and make more viruses? Well, they do this because they're obligate intracellular parasites. Now, obligate means they have to do it. Intracellular means they have to get inside of the cell. And they're a parasite because they just ultimately will destroy the cell. Okay, and so these guys, this is what makes most of them pathogenic, okay, in this sense that we're studying. Some comments about them: they do have a nucleic acid. They either have DNA or RNA. They do not have both. They have one or the other. We call that their nucleic acid core, and it's going to be protected by a protein coat. This protein coat is known as its capsid. Now, capsids can have different shapes, which gives you the different types of bacteria that you see here in this picture. We also see that it requires a host cell for them to replicate in, and this is again why they are considered non-living. Now, they do not have a plasma membrane. They do not have a cytoplasm. Okay, they do not meet the criteria as defined as living. Our next groups we're going to look at are the bacteria and the archaea. Now, bacteria, those guys can be pathogenic. They are not all, but some of them are, which means, again, they can make us sick. But archaea doesn't have any in that category. Okay, archaea has no pathogenic versions or organisms. So let's look at the cell type. Both of these are going to be prokaryotic cells. Prokaryotic cells means that they do not have a nucleus. When we look at organization, they are also going to be unicellular. They are single-celled organisms, okay? So this is where they have some similarities still. When we look at nutrition, we do see that bacteria are mostly heterotrophs. There are a few exceptions, but most of them are going to be heterotrophs, whereas archaea are going to be autotrophs for the most part, but it does vary on how they do this. So some comments about bacteria that we want to look into is that they do have some basic bacteria shapes. These are going to be things like bacillus, coccus, and spiral or spirillum, and you can see them here in the picture. They do have a cell wall made out of peptidoglycan. This is like a protein sugar mixture, and they reproduce by something called binary fission. So binary means we're going to split into two, fission means split. So binary is 2, fission is to split. And so that's how they reproduce. Now, the reason archaea is now in its own group is because they are biochemically and structurally different than bacteria. Their cell walls are very different than the peptidoglycan we see in bacteria. And there's a reason for this. A lot of these archaea bacteria, they thrive in extreme environments that you wouldn't normally find any other living thing, like the bottom of volcanoes, the bottom of the ocean, in Antarctica where it's super cold and there doesn't seem to be any food sources. These guys can live in those extreme environments. Some examples are the methanogens. They are going to produce a lot of methane gas. We do see that there are halophils. These guys love salt concentrations, so high salt. Thermophils are the ones who love extreme heat. Okay, they like really, really hot environments. And none of these are considered pathogenic. Okay, so that's the two groups that were separated from the kingdom Monera. They're now the domain that we call bacteria and archaea. So now let's get into the eukarya groups. Now these are the kingdoms, and we're going to look at Protista first. Now Protista has two kind of categories in them that fall into this microorganism group. They have the protozoa and they have the algae. The protozoa are the ones that have the pathogenic forms. Algae does not. Now, it doesn't matter what kind we're looking at, they are going to be eukaryote cells, okay? So they have eukaryotic cells. Their organization is mostly unicellular. Now, again, there are some exceptions because when we look at algae and you look at seaweed, obviously that's a multicellular organism. Okay, so there are some exceptions to the rule here, but most of them are unicellular. Now, if we look at protozoa, they are definitely heterotrophs, whereas algae are the autotrophs. And when we look at protozoa, another thing is we term them as being kind of animal-like. And the reason they're considered animal-like is they are heterotrophs, they eat their food, but also they move. Okay, so protozoa, a lot of them have the ability to move, and they can move by what we call pseudopods, which are false feet, where they can pull themselves forward. Okay, they can have flagella, which is a tail, which allows them to swim, or they can have these little hair-like extensions all over their organism or cell that are called cilia, which helps them bind and kind of crawl in a sense. In the algae group, we do see that it is going to be an autotroph. They are going to be able to be self-feeders using specifically the sun. Most of them are photosynthesizers. They are going to be able to do the process of photosynthesis, and there are some multicellular forms in this group. However, a lot of algae are still single-celled organisms. So this is the group Protista. In the group fungi, we do have some pathogenic versions of the group fungi. In this particular group, the cell type is also a eukaryote. Organization is they are mostly multicellular. Now again, exception, yeast. Yeast is a unicellular fungus. Now nutrition, they are heterotroph. They are going to actually consume their own food, but they do this through absorption. So they liquefy their food and they kind of just suck it up almost like a sponge in a sense. Now, there are two forms that fungus can come in, and there's the mold form, which is the multicellular form, and then there is the yeast, which is the unicellular form. All right, and there's a picture here that kind of shows you both forms. Last but not least, the one we want to talk about is going to be in the kingdom Animalia. Now, there are a few of these that are considered pathogenic in the kingdom Animalia, and they are normally in the group of what we call the helminths. The helminths are the worms. Okay, so there are some worms that are considered in microbiology, and they are going to be disease causing. And their cell type is also eukaryotic. They are multicellular, and they are going to be heterotrophs. And when they are those heterotrophs, they are going to be taking nutrients from us, which means that they're hurting us. Now, when we look at this, there are going to be microscopic forms of these, but in some of their life stages, they can be large, like you see here in some of these pictures, like the Platyaminthes, the flatworm, and the nematodas, which are the roundworms. They can be actually very, very large, and you can see them with your naked eye. All right, but we are going to dive into more detail about each of these groups as we go through and talk about the different types of microorganisms. So this finishes up part 1, where we're just doing our introduction. Part 2 of this introduction is going to focus on the history of microbiology. Where did it come from? What scientists made great advancements in it? And so that's what we're going to be looking at in part 2. If you have any questions, please let me know.